Hello everyone, welcome to sBiology. Previously, we have discussed about the Watson and Crick model of DNA double helix. Today, we are going to discuss about the experimental proof for semi-conservative DNA replication by Meselson and Stahl. So, before going to the experiment, that is Meselson and Stahl's experiment, we have to know what is semi-conservative mode of DNA replication and what is DNA replication. Okay. In addition to the double helical structure of DNA, Watson and Crick also proposed a scheme for DNA replication in 1953. When Watson and Crick proposed the double helical structure of DNA, at that time they proposed a scheme for DNA replication in 1953. Okay, according to this scheme, the two strands of double helix would separate and act as template for the synthesis of new complementary strand. जेतेबेले वाटसन एंड क्रिक डीएनए डबल हेलिक्स मॉडल प्रपोज करथिले ठीक सेही टाइम रे से गोटे स्कीम देथिले कहा पाई कि ना डीएनए रेप्लिकेशन पाई से कोन कहितले ना the two strands of double helix would separate and act as template for the synthesis of new complementary strand. से कहितले जेतेबेले डीएनए रेप्लिकेशन होउची प्रथमे जो पेरेंटल डीएनए अछि से कोन होउची फर्स्ट सेपरेट हे जाउची तो डबल स्ट्रैंडेड डीएनए फर्स्ट सेपरेट आउट. ओके, two strands of a double stranded DNA would separated out first. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme for DNA replication was termed as semi-conservative DNA replication. Okay, we have already studied that replication is a biological process by which two double-stranded DNAs are formed from a double-stranded DNA. Okay. But in case of semi-conservative mode of replication, when two daughter strands are formed, they contains one parental strand and one newly synthesized strand, according to Watson and Crick scheme of DNA replication. तो DNA replication पाई कि वाटसन एंड क्रिक जो स्कीम प्रोड्यूस करथिले से कहिथिले जे जेतेबेले रेप्लिकेशन कंप्लीट हे जाउची जो दीटा डॉटर डीएनए सृष्टि होउचंती से माने कोन करूचंती गोटिए ओल्ड स्ट्रैंड गोटे पेरेंटल स्ट्रैंड एंड अनदर न्यूली स्ट्रैंड कंटेन करूचंती. तो से जो प्रोसेस टा को कहा जाउची सेमी कंजर्वेटिव mode of DNA replication. Semi means half. The term semi means half. Okay. Conservative comes from the word conserve. Okay. So half strand from old or parental and half strand from newly strand. Half strand is new and half strand is parental or old. That's why it is known as semi-conservative DNA replication. Half or semi type or semi DNAs are conserved. Okay. So, Watson and Crick proposed the semi-conservative mode of DNA replication, but they did not able to design any experimental proof for semi-conservative DNA replication. But in 1958, Matthew Meselson and Franklin Stahl proposed experimental proof for semi-conservative DNA replication. Okay, Meselson and Stahl in 1958 proposed an experimental proof for semi-conservative DNA replication. What they did, let's see. Matthew Meselson and Stahl first grew E. coli bacteria in N15 medium, that is N15 ammonium chloride. Then what is N15? N15 is a heavy isotope of nitrogen. N15 is a non-radioactive heavy isotopes of nitrogen. Okay. After that, all cells all E. coli cells accumulate the heavy nitrogen as a source of nitrogen. Sorry. Okay. So all the molecules having nitrogen including DNA became heavy due to the heavy isotope, that is N15. Okay. After that they, Meselson and Stahl, extract the DNA sample from these cells and subjected it to cesium chloride density gradient centrifugation. Then what is centrifugation? Centrifugation is a separation process, and they can easily detect the heavy DNA from the normal DNA because the heavy DNAs are present or found on the bottom of the test tube due to heaviness. Okay. In comparison to the light DNA, if the media will contain the light isotope, that is N14 ammonium chloride, then the DNA will appear on the top portion of the test tube here due to light in comparison to the heavy DNA. Okay. So after some time they transfer the E. coli bacteria from N15 medium to a fresh N14 ammonium chloride. Then what is N14? N14 is a light isotope of nitrogen. Okay. And the bacterial cells, E. coli bacteria, can multiply, can doubles in each 20 minutes. Okay. E. coli bacteria गुड़ाक प्रत्येक 20 मिनट रे थरे डबल हो जाती. डबल मीन्स they reproduce in each 20 minutes. गोटे E. coli bacteria रू दुईटा दुईटा रू चारिटा एते हेई कोन होती डबल. They multiplies in each 20 minutes. Okay. So after 20 minutes, Meselson and Stahl again collect the DNA sample from this N14 ammonium chloride and again subjected it to cesium chloride centrifugation, and they found that the DNA's band are appeared on the mid portion of the test tube, not in bottom or not in top. They found the DNA bands on the mid portion of the test tube. Why these DNAs are appear on the mid portion of test tube? Because these are hybrid DNA. Okay. One strand contains heavy nitrogen N15 and another strand contains light nitrogen from light ammonium chloride. Okay. As they are growing on the media containing light ammonium chloride, the one strand from parental type and another strand from newly synthesized type, that is N14. Okay. So in the first generation they observed that hybrid DNAs are formed and appear on the mid portion of the test tube. Okay. Then again after 20 minutes, 20 plus 20, 40 minutes means second generation. I said that E. coli can doubles in each 20 minutes. In the first 20 minutes, they collect the hybrid DNA. Okay. Then again after another 20 minutes, after 40 minutes, they again collect the DNA samples and subjected it to cesium chloride density gradient centrifugation. And in the second generation, they found that some DNAs are present on the mid portion of the test tube and some are found on the top of the test tube. And the top DNAs are the light DNA having the N14 DNA and the mid portion DNA are the hybrid DNA having both N14 and N15 in the second generation. बुझिला किछि प्रथमे मेसेलसन एंड स्टाल कोन कले किछि बैक्टीरिया नेकिरी गोटे मीडियम जोटा होची हेवी नाइट्रोजन अमोनियम क्लोराइड सेठी ग्रो करिके कोन करिले ना तार डीएनए को एक्सट्रैक्ट करले एट 0 मिनिट बिफोर द रिप्रोडक्शन of bacteria. बैक्टेरिया रिप्रोडक्शन करने पूर्व तार कौन कले डीएनए को कलेक्ट करने कलेक्ट कर सारिकी कौन कले सेंट्रिफ्यूगेशन प्रोसेस करले विथ सेसियम क्लोराइड डेंसिटी ग्रेडियंट से ऑब्जर्व कले जे डीएनए गुडाक टेस्ट ट्यूब रो तळे बा बॉटम रे रहि जछन काहे कि ड्यू टू द हेवीनेस heavy DNAs are easily detected from the normal DNA as they are settle on the bottom of the test tube बुझेला तो तापर कोन कले से कोन कले ए जो डीएनए बा ई कोलाई गुडा को रहिथिले हेवी मीडियम रे ताको ट्रांसफर करले गोटे फ्रेश लाइट अमोनियम क्लोराइड व्हिच कंटेन्स द N14 हो जोटे लाइट आइसोटोप ऑफ नाइट्रोजन तो एठी कोन रे ता ट्रांसफर करला परे से ताको 20 मिनिट छाडी देले बिकॉज़ द बैक्टीरियाज आर डबल्स इन ईच 20 मिनिट्स तो 20 मिनिट परे से कोन कले पुनी आउ थरे डीएनए को कलेक्ट कला परे से कोन कले पुनी आउ थरे सब्जेक्टेड टू सेसियम क्लोराइड सेंट्रिफ्यूगेशन डेंसिटी ग्रेडियंट सेंट्रिफ्यूगेशन सेठी देखिले से जे जो डीएनए डबल स्टैंडर्ड बैंड गुडा को अपीयर हेले कोटी मिड पार्ट ऑफ द टेस्ट ट्यूब एंड दीज आर द हाइब्रिड डीएनए ओके एंड आफ्टर again another 20 minutes after 40 minutes they again extracted the DNA collect the DNA and again subjected to the cesium chloride density gradient centrifugation and they found that some DNAs are appear on the or present on the top of the test tube and some are on the midpoint of the test tube because some are light DNA and some are hybrid DNA काहेकि ना जेहेतु आमे कोन करथले ना लाइट मीडिया रे ग्रो करैथले डीएनए गुडा को कहला किछि ड्यू टू द प्रोसेस ऑफ सेमी कंजर्वेटिव रेप्लिकेशन सम आर लाइट एंड सम आर हाइब्रिड ओके केमिति होची देखिबा आसो लेट्स सी हाउ इट पॉसिबल इन द फर्स्ट जनरेशन और इन द जीरो जनरेशन when they grew the E. coli on the heavy medium and the collect the DNA. They found that the DNA is heavy, having two strands are N15 and N15. Okay. In the first generation, when they transfer it to the N14 media, in the first generation, the parental DNA strand, we have studied that. At the time of replication, the parental double stranded DNA would separate first. Then first, these two strands are separate out. This is one strand N15 and this is another strand N15. After that, after the completion of replication, one newly synthesized DNA attached to the old DNA or pair with the old strand of DNA and in the first generation form hybrid DNA. In the first generation hybrid DNAs are formed. Okay, one strand is parental type and another is newly synthesized and having N14 because they were grew in the media N14. Okay, after the second generation, again these strands are separated out. First strand will separate one blue and another red. One blue, another red. Blue is the red is the daughter or new strand and a blue is the parental strand. Blue is parental strand. Okay. So again these two DNA separated out. Okay. This DNA separate as blue, red, and this DNA also separate as blue and red. Okay. And another newly strand are pair with the parental type. And in the second generation, they found that 50% are hybrid and 50% are newly synthesized. 50% having both N15 and N14 and 50% having only N14 in the second generation. Then after the second generation, after third generation, they found that 75% of the DNA contains only light DNA, and the hybrid DNA amount is decreased because they grew on the media N14. Okay, and this is the semi-conservative mode of replication. Okay, so this is all about the Meselson and Stahl's experiment of semi-conservative mode of replication. In the next video, in the next class, we will discuss about the process of semi-conservative DNA replication. Till then, keep watching and keep learning. Don't forget to subscribe my channel, that is sBiology. Thank you.